Bismillahirrahmanirrahim. Assalamu alaikum and good day, ladies and gentlemen. I'm Hazima Mozam and I welcome you all to the 6th lecture under COMSTECH NDT joint lecture series. We are very grateful and to Professor Dr. Ehmke Pohl. Professor Dr. Ehmke Pohl is a full-time professor at Biophysical Sciences Institute at Durham University. The title of his talk today is Structure-Based Drug Design Towards the New Anti-Infectives. Professor, we are very, very grateful for your time and for your kind acceptance. With this, I would request Professor Dr. Iqbal Chaudhry, Coordinator General COMSTECH and Director ICCBS, to welcome and introduce our guest speaker for today.
Assalamu alaikum, good morning, good afternoon, and good evening, because there are people from extreme west to the extreme east. There are 4 continents in which we have membership. COMSTECH is a standing committee for science and technology with 57 member states. So my dear Professor Ehmke Pohl, you would be actually addressing scholars from different countries of the world, and they are in different time zones. So without doubt, I think they are some of the brightest people because they have a lot of interest in science and contribution of NTD in developing their understanding, appreciation, and capacity in the field of tropical diseases, neglected diseases are highly appreciated. I met MK some 4 years ago. I was in Durham and we had this opportunity 2 years ago also. We had this opportunity of meeting with each other, and I was completely and fully impressed from his commitment, his excellence, and his vision for global science. He has been instrumental in initiating many projects in many countries of the world, including India and Pakistan and other countries, including in South America also. He is truly a scientist with a great commitment for a global good. Professor Pohl graduated in chemistry at the University of Göttingen and finished his PhD in 1994 with thesis on bioactive peptides with Professor G.M. Sheldrake, a very famous name. He continued his training in protein crystallography as Ernest Schering Fellow at the University of Washington in Seattle, USA. In 1999, he joined European Molecular Biology Laboratory outstation in Hamburg as an independent scientist and team leader. In 2004, he took up a post as principal beamline scientist at Swiss Light Source, responsible for the Max Planck Society, Novartis, Hoffmann-La Roche beamline. In 2007, he moved back to academia to take up a position as leader in a joint appointment by Department of Chemistry and Bioscience at Durham University, UK. In 2015, he became co-director of the Biophysical Science Institute at Durham University, where he pursued his interest in combining biophysical instructional method with structure-based drug design to combat infectious disease. Most profoundly, he has used his wonderful skill of structural biology by using X-ray crystallography to solve some of the most enduring problems in tropical disease, especially parasitic diseases. He is a scientist of excellence. He's a good speaker. At the same time, he's always there to help younger people. So he has this great capacity to mentor younger people, and that make Professor Ehmke Pohl a very unique individual, as someone with whom a lot of benefit is coming to the world and to the young people. With this, I would like to invite him for his presentation.
I'm blushing. Thank you so much. Far too much honor, but I want to start by really thanking you guys for inviting me. This is just amazing. this talk series, and the idea that we can really sit all over the world and share ideas and yeah, share our science. It's absolutely fantastic. And I do remember our dinner 2 years, almost 2 years, a bit more than 2 years ago. And it was just such a wonderful evening meeting you and having a global perspective on the science that we do. And I think a lot of us share this vision that we do science to actually benefit people and benefit humankind. And I don't think anybody's here for the money. So thank you so much. I'm going to start sharing my screen now, if that's all right. So all right, does this work?
Yeah. Yes, it's working.
Okay, so I was asked to talk about structure-based drug design towards new anti-infectives. Can you hear me all right?
Yes, it's loud and clear.
Good. So I thought I'm going to start with something somewhat differently, and this is really a reminder on the there we go. The reminder of the power of structure-based drug design, because obviously over the last 2 years, COVID-19 has really changed our view on international science and on infectious diseases. And I just want to sort of remind people that the most promising anti-COVID drug, Paxlovid, that was just approved recently and developed within less than 2 years, is almost entirely based on structure-based drug design. So I just wanted to sort of remind people that if we actually work together as a community and as a worldwide effort, and Pfizer is behind it, we can actually make drugs in a surprisingly fast way. Now, I'm not going to talk about Paxlovid as a drug, but I just want to also mention that this development was really greatly aided by all the structure studies. And these structure studies actually started 20 years ago. So on the one hand, this drug is an example of how fantastic structure biology is and how it really can aid drug design. But on the other hand, it's also a reminder how much long-term effort is required in order to come to this point. And even this seems to be quite a quick development in 2 years. It's really based on 20 years of research at Pfizer and actually many other groups working on closely related proteases. So what I'm trying to do in sort of my talk is giving a bit of an overview how we as a community approach structure-based drug design and where we can actually support other studies. So I'm going to start with sort of the easy introduction on how, in principle, we visualize the structure-based drug design approach. And of course, the first point is always target identification. And here, the NTD network has actually done quite a lot of work in order to identify new targets. So if we are lucky and work hard, we might be able to get crystal structure relatively quickly and have three-dimensional structure at high resolution at the beginning of the process. So from there, we're often trying to look at protein ligand crystal structures in order to identify the atomic detail of our interactions. And then as soon as we have the crystal structure, we can also start in silico screening. So we use computer power in order to find new ligands. And more recently, fragment-based screening, or more recently, meaning over the last 10 years, fragment-based screening has become a very popular high throughput technique in order to find our first protein ligand crystal structures. And once we have that, we basically enter the so-called drug design cycle, where we use then biophysical and biological assays to look at the efficacy of our ligands, and use further in silico docking to optimize our ligands, then go over to our synthetic chemists next door and give them hints on ligand synthesis. So in my view, crystal structure gives in the aid the ligand synthesis in many ways. And that, of course, goes back into protein ligand crystal structures. And after a few rounds, if we are lucky, we come up to further development with a structure and a compound that is close enough for clinical trials. So if you sort of keep this very simple and really simplified view, in reality, of course, life is much more complicated, but if you keep this simplified view in mind, I will try to walk you through the steps that we have undergone in one particular project in order to come from target identification to ligands that are in principle ready for further analysis. Now, the example that I've chosen is actually tuberculosis, which is not one of the neglected tropical diseases, but of course, as many as you know, is still one of the world's top killers. In fact, the WHO has realized that there's a huge relationship between tuberculosis and COVID-19. Because of the COVID-19 pandemic, much fewer people receive TB care. The WHO estimates that there's an additional 500,000 deaths on tuberculosis due to COVID-19. So just very quickly, the WHO really calls for better effective prevention and control, but also for some simultaneous testing of TB and COVID-19. I think the last thing is actually important. The WHO also calls for substantially increased investment in TB research, because certainly one thing that the pandemic has shown us is that we really are only reacting to the disease, and there's very little proactive research going on. So I would like to support the WHO on this and really say we do really need more investment, not just in TB research, but in infectious diseases. So in principle, tuberculosis treatment is based on a number of relatively old drugs that have been on the market for decades. So for normal tuberculosis, you would normally get a cocktail of 4 drugs for 6 months, including this little compound on the right-hand side called isoniazid, or INH. Multi-drug resistant tuberculosis, which is of course on the rise, and extensively multi-drug resistant strains require a cocktail of at least 6 drugs for 2 years. So you would normally add cycloserine and ethionamide, ETH, which is shown on the right-hand side as a drug. So the problem with these cocktail of six drugs is that they produce significant side effects. And often people stop taking the drugs too early, and then tuberculosis reoccurs. So we and other people have been looking on ways to increase the efficacy of these drugs. Now, very quickly, ETHA and INH, they're both pro-drugs that are actually converted by MTP enzymes into reactive NAD adducts, shown down here. I'll get a laser pointer. There we go. So these drugs are actually being converted into its active form by these tuberculosis proteins. So if you look at these small product molecules, they're really small molecules that actually can penetrate the sturdy cell wall of tuberculosis. And then ingeniously, they're only converted by mycobacterial proteins into active forms, which then inhibit the fatty acid synthase structure. And the target that we have been looking at is called ETHR, which is actually the transcriptional regulator that regulates the bioactivation of ETH, the enzyme of ETHA, the enzyme that is converting the drug into its active form. And about 10, 15 years ago, people have realized that if we inhibit ETHR, the regulator, we increase the expression of ETHA, the molecule, the protein that activates the prodrug into its active form. So basically, an inhibitor of this protein ETHR acts as a booster drug for ETH. And that's a very convenient or very, very clever way, because what we actually do is we increase the efficacy of a drug that is widely used, widely known, cheaply available to everybody. So the idea is that with these ETHR inhibitors as a booster drugs, they would simply be added to the cocktail that is being given for multi-drug resistant tuberculosis cases. So obviously this was done work before we joined the project. We actually came onto this project when people started looking at the structure of these proteins. The reason is that ETHR is a member of the TET-R family. So this is a picture of one of our TET-R proteins here that's shown in ribbon diagram. So we have a TET-R protein called VCER, which is bound to DNA, a structure that we haven't published yet. And the people who were working on ETHR basically contacted us and said, you have some experience in TET-R repressors, why don't we collaborate? So TET-R repressors are generic bacterial repressors that are often involved in repressing genes that are involved in drug resistance. So again, this is an example of the VCR repressor from Vibrio cholera, and that actually regulates the efflux pump system of Vibrio cholera. So the advantage of these proteins is that they're unique to bacteria, so there's no eukaryotic equivalent, it's less likely to have any side effects. And secondly, they're widely distributed over bacterial species. So it's quite likely that inhibitors of these proteins can actually work for several strains. So what you need to remember is that we're actually looking for ligands and inhibitors that prevent DNA binding, because once they the ligand binds, the ligand inhibits the DNA binding activity, the DNA becomes available, and the genes are being expressed. So almost, 16 years ago, then the group in France who was working on this solved the first crystal structure of ETHR, which is shown up here. So it's a crystal structure of ETHR just with the ligand, and it's the generic ETHR or TET-R repressor, the homodimeric protein with the DNA binding domain up here. So we have two DNA binding domains up here and a ligand binding domain down here. And as you can see in the ligand binding domain, they found a molecule that's probably something like a fatty acid, although it's never been really proven what the natural ligand is for ETHR. So this was the point when they actually contacted us, because as I said, we had some experience with DNA binding proteins and then TET-R repressors. So we started out and thought, okay, how can we use structural information to find further inhibitors? There's one thing I should mention, and the expert, the structural biology expert probably sees that. If we look at the conformation of these DNA binding protein, we can see that this DNA binding domain and this DNA binding domain, they're basically turned around by 90 degrees. So they are in an inactive conformation. So basically, when they solved the first crystal structure, they were a bit lucky because they solved the structure with an inhibitor bound in a conformation that is not compatible with DNA binding. So our starting point is, this is the conformation we want. And all we need to do is we need to find new ligands that basically force the protein into the same inactive conformation. So when we joined the project, there were already 12 ETHR complex structures in the protein database. And the first thing we did is we looked at the different structures and superimposed all the structures. And as you can see, they look almost identical. In fact, through most scientists, they would look completely identical, and people would think, we don't learn anything from those crystal structures. But if we do look at the different binding sites, and in fact, this is sort of the binding sites in the different crystal structures in different colors, we can actually see that there are quite significant subtle differences between the different crystal structures. So although these 12 crystal structures on the first view look identical, it is the subtle differences up here that actually are important. So the small side chain movements up here, and in particular up here, I'm going to come to this problem a little bit later, seem to be absolutely relevant. So at this point, we then started, okay, we have all these high resolution structures. How do we come into a in silico screening to our next step? So the basic idea was that rather than docking all of our potential ligands against 12 structures that might be very similar, which would be huge computational cost, we try to identify the best protein structure for docking using a cross-docking experiment. So I'm going to explain this in silico cross-docking experiment in a moment. So if you think about the computational time, you really reduce the computational time by more than 1 order of magnitude by choosing the best protein structure and not choosing all 12 protein structures. And at the same time, if all the protein structures are similarly successful for docking experiment, you don't want to waste your time on docking again and again against the same crystal structure. The second thing that we needed to do, we also needed to define a subset of potential ligands for our virtual screening in a pre-screening protocol. So again, we have databases of millions and millions of compounds. And if we can rather pre-screen those millions of compounds to a more manageable number, basically your individual docking can be more sophisticated. If you use the entire chemical space, all available chemical compounds, you will again waste a lot of time docking compounds that are very unlikely to be in any ligands. Nevertheless, we had to actually resort on some supercomputing power down in Cambridge in order to screen a large number of potential ligands. And then we took those best ligands for further identification, biophysical, and structural validation. And I'm going to show you a few examples of that at the very end. So this cross-docking experiment was actually really crucial. I'm going to show you why. Because what we basically did is we basically took our 10 best ligands in 11 structures, and then we hit those into a decoy data set of about 350 ligands that look like ligands, but they're not ligands. So basically we hit the true ligands in 350 potential ligands, and we were then trying to find the best crystal structure that actually finds the known ligands on top of the list. And it turns out that the differences between all these different PDB codings are actually huge. So remember, on the first view, these crystal structures look all the same. So each of these bars represents a different crystal structure that looks almost identical. Yet there's one crystal structure that is much more successful in finding the true ligands at the top of the pile, where some of these crystal structures are really not very good. So they don't find our top structures in the top. And then there's a tiny little detail, and I always like to sort of show this as an example, how really attention to detail, attention to single atoms makes the difference. So looking at this asparagine residue down here in the binding pocket, you can see that there's an oxygen atom on this direction and nitrogen atom in this direction. So this was in all of the 12 crystal structures. Now we looked at this and analyzed the hydrogen bonding pattern and realized that this needs to be turned around. And of course, that's a tiny, tiny change because they're actually isolated electronically, but of course they do have a different hydrogen bonding pattern. So we realized that there's really the tiniest of errors in the crystal structures in the PDB, and this needed to be turned around. So the nitrogen needed to be here, and the oxygen needed to be here, right? And that had actually a huge difference on the success rate of our cross-docking experiment. So first of all, the success rate overall went up, and also the success rate for this particular PDB structure was actually on top. So, and this is something that can easily happen. You're just trying to fit the ligand to the density, and sometimes people don't take enough care looking at the exact hydrogen bonding pattern that the chemist would do. So turning this single residue around made a huge difference. And this then cross-docking experiment also told us that we really should only use this crystal structure, 1U9N, for our in silico high throughput screening. So that was, again, quite surprising to me. So the next thing we did is we looked at the crystals, at the compounds, and we used the Zinc chemical database, which is freely available. And at that time, it contained more than 35 million commercially available structures in a ready-to-dock format. So anybody can just download the entire database onto your local computer. We then used a subset of 6.1 million compounds that are classed as drug-like compounds by the Zinc database already. So that already reduces the number of molecules that we wanted to dock by a factor of 5. And then the second thing, we spent quite a lot of time on optimizing the molecules out of these 6.1 million that actually can fit to our binding pocket. So we used something called a NIME pipeline for that. I'm not going to go into detail. I just want to show you a few sort of highlights that prove really important. So because we know quite a lot about our binding pocket, we can actually find molecules and pre-screen molecules that are more likely to bind our binding pocket than others. So for example, we know what the volume of our pocket is. So we kept 200 to 700 cubic angstrom, which is a relatively small volume. But we also wanted to have smaller compounds because we wanted to leave space for further chemical synthesis. So we deliberately took the decision to look at small compounds that could be starting points for further chemical analysis. In fact, we knew that the largest pocket that we found is actually 1200 cubic angstrom. So quite a lot of space. Another thing that we wanted to see, we really wanted to explore new chemical entities. So we wanted to have a number of ring atoms, rings. So we wanted at least 2 ring systems in our structures. And there's a few other criteria that you probably recognize in terms of molecular weight, hydrogen bond acceptor, and so on and so on and so on. So this really then brought the number of compounds down quite about to about 1.3 million. So again, so now we are another factor of 5 lower in terms of the number of structures that we need to dock. Now, because there were already people working on ligands, we really wanted to expand our chemical space. So without going into too much detail, there's a bit of a larger 9 document that really clustered our compounds into different chemical entities. So there's a huge pipeline where we basically cluster all of the remaining 1.3 million compounds according to chemical diversity and according to different ring systems. And that actually left us with 400,000 compounds. So 400,000 compounds is 2 orders of magnitude fewer compounds than actually are in the original Zinc database. So this pre-screening, which probably took about 6 months of work, really reduced the number of compounds that are less likely to bind. So this was then put into a supercomputer. We used a program called Gold for docking, which was developed by the Cambridge Structure Database because we were collaborating directly with them. And we figured on our computers, this would have taken about 56 weeks. But again, we were lucky to get some time on a supercomputer in Cambridge, and the entire docking took only about a week of computational time. So that was actually quite convenient. But we still now have 400,000 compounds, and because Gold uses a genetic algorithm, each of these compounds has 5 different binding poses. So we're basically looking at 2 million different binding poses. So we needed to find the post-docking algorithm to come down to the compounds that are actually useful. So first of all, we can take the top 10% of all solutions just based on the Gold scores, so the score of the dockings. Yes, and I should mention, I forgot to mention, out of our 400,000 compounds, only 6 compounds didn't find any solution whatsoever. And that is testament to the pre-screening protocol, where we already pre-screen all those molecules that are less likely to bind because they are way too big or way too small or way too hydrophilic or way too hydrophobic. We've already taken those compounds out. So this leaves us with about 40,000 compounds, and we still need to bring this down to a manageable number. So what we can do then is we can then take all of those compounds and use both the physical, chemical, but also the structural descriptors to reduce the compound. So basically, we can look at the now docking pose and only take the best of the docking poses. Now, there's a number of these structural descriptors, and I'm not going to go through all of them, but I'm just showing you an example. So this is, for example, a value for how well the ligand fits into the pocket. So it's the ligand clash count. And basically, we can say, okay, we only take the ones that have the lowest ligand clash count. So all of the compounds that have a large number of clashes, we take them out. And then we can look at the hydrogen bond donors. And again, the ones that don't satisfy the hydrogen bond donors, we take them out, and so on and so on and so on. So what is important is that we have about 20 of those descriptors, and we have to use all of them in order to come down to reasonable numbers. There's not a single descriptor that rules out a lot of the compounds, but every descriptor rules out some of the compounds. And only by really combining all of the descriptors and using all of the information, we can actually come down to a number that is manageable. So 300 molecules is actually manageable. We can look at 300 molecules on the screen. So at this point, my PhD student, and I'm going to show her picture later, Natalie, came up and said, so what are we going to do next? And I said, well, we use the most powerful tool in the universe, and that's our human brain. You look at each of these 300 structures and basically make an assessment if this is a binding that makes sense or it doesn't make sense. This sounds a bit unscientific, but it's been shown again and again that we do have something, some sort of intuition, and looking at structures and from different angles, we can actually see that, well, this structure makes sense, or this ligand is really too distorted, or this hydrogen bond doesn't fit, in spite of all of those descriptors. And this is really very, very powerful. And again, I'm going to show you a bit of an example for that up here. So these are two of the compounds, and these are actually quite similar chemical entities. And so we could basically decide we don't really need to buy both of these chemical compounds because they are quite similar. And just looking at this from different angles and turning this around, we could see that actually this one looks a bit better than this one. So we took this one out. And there were a few docking poses that were clearly, in spite of all the clever computational stuff that we have done before. So there were a few of those that were clearly not very good. So we just checked them out. So basically this ended then up with about 85 compounds. So remember, we started with a Zinc database of 35 million, got this down to 6.1 million by using the drug-like compounds, got this down to 400,000 by using the chemical descriptors, something that fits, is more likely to fit into our binding pocket. Then comes the docking with all the post-docking screening. And at the end, we come up with, about 85 compounds, which is simply a manageable number of compounds. And because this is based on commercially available compounds, we could actually buy all of those compounds for a relatively small amount of money and test them. So the first and easy test that we could do is called a Thermoshift analysis, which is shown up here. And the Thermoshift analysis is based on a very simple premise that we basically heat up our protein with a bit of the compound, and we measure the unfolding of the protein with a chemical probe. And so its melting temperature of the protein is supposed to increase when a ligand is bound. So again, it's a very simple premise. We simply assume that if a ligand is bound, our protein is more stable because the ligand holds everything together. So we increase our melting temperature, and we can do this in a 96-well format, with a simple PCR machine. And it turns out that out of the 85 compounds, about 20 compounds had a significant shift in thermal shift, thermal shift. So these are the thermal shifts at different concentrations for the different compounds. And you see in particular, this one up here has a thermal shift increase of thermal stability by 8 or 9 degrees. This one is also very nice, goes up to 7 degrees and so on. So the success rate of about 20 of molecules out of 85 is actually a very high success rate. So we talked to our friends and collaborators in Cambridge, and they were quite surprised. Normally, they said you would be happy having 1 or 2 high hitters. So this is really a testament to the fact that the pre-screening worked really well. We pre-screened and docked the molecules quite well, and ended up with 20 potential candidates. So being a structural biologist and having the protein in our hands, and in fact the crystallization conditions were known, we then of course started with crystallization trials right away. And again, I'm just going to show you a few examples up here. So this is one of our compounds that, this is one of our compounds solved at a resolution of 2.1 angstrom. Crystals are always very nice up here. And it's exactly bound in the binding pocket as we've seen. So again, this is just showing the monomer of the structure with the DNA binding domain up here, the ligand binding domain down here. And as in the previous structures, this is in the inactive conformation. So the ligand holds the protein in its inactive conformation. This is the the electron density, and the few crystallographers among us, Ashish, will probably realize that looks really pretty. Now, one thing that was particularly nice, a lot of the compounds were somewhat colorful, and we could actually see that the crystals that were soaked with the compounds also adapted the same color. So again, from an experimental point of view, this was a very satisfying project because the crystals grew nicely, and you could really see how the compound would bind into the crystal and really be soaked up. So with that, we could then compare the docking pose versus the true structure. And we've done this up here. This is basically a superposition of the best of the different binding poses with the X-ray structures. Remember, because we're using a genetic algorithm, we get 5 different binding poses for every single ligand, and they're sorted according to their score. And it turns out that the highest score is sort of quite okay, maybe not perfect, but quite good. Not perfect up here. Second one, the ligand is turned around by 180 degrees. And it's the third one, which is actually really very similar to the X-ray structure. The fourth one and the fifth one, again, really not correct, with the binding pose turned around. So this basically shows that although we use, I would say, a very sophisticated docking algorithm, the docking score alone is not sufficient to find the true structure. So again, our best docking score is this one up here. So if we had relied on the docking score, we would have said this is our binding pose. And in fact, this is one of the examples where the docking poses are quite good. In other examples, the docking poses are actually not as good at all. So for me, this is just a sort of reinforcement is that we really need structure validation by an experiment or more sophisticated computation. Docking alone gives you a docking pose, which is a good guideline, but it's usually not good enough to guide the chemistry guide the chemistry in the next step. So obviously, we also wanted to check how well do these compounds work. So we sent them to our collaborators at the Institute Pasteur in Lille, and they have a growth assay in E. coli based on GFP fluorescence, and they have an experiment where they basically check how much does our compound increase the efficacy of ETH. So this is done at the 20th of the normal concentration that you would use for ETH, and you simply look at the efficacy of this compound. And I've put this up in this table compared to the delta TM. So you see number 10, compound number 10 has a decent delta TM, so it definitely binds to our ligand, to our protein, but it has no efficacy whatsoever. 25, again, is one of our top compounds. It has a very good efficacy. But we also have number 57, which has no efficacy whatsoever. So 57 was one of our top hits in our Thermoshift assays, but again, no efficacy whatsoever. And the best explanation that we have is that this is probably relatively large and not able to penetrate the cell wall. So that tells us that in our pre-screening, we really have not been able to take the biological knowledge into account. So we only looked at physical, chemical parameters, and we didn't look at parameters that would actually determine how good the uptake is. And I think this is one of the steps where we, in the future, hopefully can be better, that we use those biological data earlier on in the docking process. Now, again, this is the best one, compound 25. This is the smallest and the most promising lead compound. In a sense, it's really nice. It's almost as small as a fragment, which means that it leaves us a lot of space for further chemical synthesis. So this is where we went to my colleague Matt Kitching and a master student who is now doing a PhD, Callum, and said, why don't we make a few more compounds? So based on the crystal structure up here, we could see where the ligand is bound. And I mentioned this binding pocket, which was on the top of structure before. This is actually this free space up here. And in fact, we knew from other crystal structures that this phenylalanine can move around and adopt a conformation on this side. And that really opens up this entire pocket as a very hydrophobic pocket. So you can see there's a couple of hydrophilic interactions with these asparaginines up here, with the amide bond in the center. But here we have a lot of space for further compounds. So consequently, he made all of these compounds. We measured the TM, and you can see there's a maximum in terms of chain length in, up here, where we can still bind to the pocket. If we increase the chain length any further, the melting temperature goes down, so the binding goes down. And it's probably that we're not filling the binding pocket perfectly, and we might sort of push the entire ligand more into this direction. So again, based on the crystal structure, we could change this part. And then we did a few more. So basically, all of these compounds and started with the structure-activity relationship based on the Thermoshift assay. And as you can see, our best compound right here has a delta Tm of 9.9 degrees. So this is the highest delta Tm that we have seen. And you can also, of course, do a bit more chemistry on this side. So again, the important lesson is that the crystal structure really could support and aid where we can change our synthesis. Now, at the same time, we were also sort of looking into how can we improve the process. And in terms of computational studies, MD simulations would be, full MD simulations would be the next step. So when we started with these MD simulations, there are more than 70 crystal structures out there, and there are hundreds of compounds available. Now, we've already been shown that the simple docking with gold is not good enough to really reproduce the crystal structure in most cases. So the question is, if we use more sophisticated molecular dynamic simulations and use calculations of free binding energies, can we get closer to this? And we looked at the number of crystal structures that actually had multiple binding modes. Because one of the challenges in crystallography is sometimes the binding, the ligand is not very well defined, and we have to model it with two binding poses. And we really don't know which one is the most likely. Is it just a superposition? Is it just disordered? So maybe MD simulations come a bit closer. So this is one of the examples where we actually have a compound which is on a somewhat unusual binding site. So we see this in a moment up here. So this is actually superposition of our best binding pose identified by docking with our crystal structure. And we can see that this fits actually quite nicely. Right? The second best binding pose is actually perfectly in quite a different direction. So what we basically see is that if we use MD simulations with the starting point of a docking pose, we can verify these. We actually arrived at the crystal structure in a much more consistent way. Now, MD simulations usually take days or weeks, so we can't do this on a large number of compounds, but we can certainly do this on a couple of compounds and really see, can we get a better grip on actual binding poses. So coming to an end of this, the end of the story is, where are we with these compounds? Now, the proof of concept basically has been done, and these booster drugs targeting ETHR have now been pursued by fragment screening in Yale and Cambridge, and in particular by extensive medicinal work in Yale together with the company. And they have actually developed this particular compound down here, which is based on a different series. And with the help of a large EU grant, they're actually putting this compound now into phase 1 clinical trials. I should say, unfortunately, we are no longer part of this consortium for various reasons. So we're working in parallel. We're still working with Nicolas and Alain at the Pasteur Institute, but this is now being pursued independently by this little company. So overall, I hope that I've shown you how we can actually use crystal structure as a starting point for a structure-based approach, go through sophisticated docking, and come up with new compounds that can be lead compounds for further development. And with that, I just want to thank a few of the people involved. There are far too many of the key people up here. So Natalie is my PhD student, Natalie Dayton. She started the whole project and did all of the computational work. Rose was a master's student who did the first set of improved compounds. Callum is the PhD student now working with Matt Kitching, who has made all these compounds, and Charlie's a former PhD student who is now working actually at Diamond. Natalie is working up in Newcastle. He is now working at the XChem screening facility. He did all of the crystal structures. And of course, Alain Nicolas from the Pasteur Institute, Jason from the Cambridge Structure Database, and Lynn and Fernanda, they help with the molecular dynamic simulations. And with that, I want to open for any questions. Thank you so much.
Thank you very much, Professor Pohl, for a very elaborate and fascinating lecture, I would say. And you have shown a lot of work, and I suppose it's an effort of so many people under your leadership. So let me see if we have questions in the chat box. There are so many comments about your presentation. Let me read out some for you.
Read them. Thank you, guys.
Yeah, thank you very much for your professional and interesting webinar. Very informative information. So yeah, we have one question from Ms. Faryal Ashraf. She's asking, hi, Professor, your talk is very informative. My question is, I would just like to know, in case of thermal shift, case of thermal shift assay or DSF that you did, you find all the compounds give increase of 8 to 9 degrees of temperature. However, in our case, we found that both kind of responses, like increase or decrease, we would like to take these results, like what could be the case when you have to increase or decrease, or if I misunderstood, then kindly elaborate. She's probably asking that the thermal assay that you have shown, it shows increase in temperature from 8 to 9 degrees, but in their case, they are also observing some decrease in temperature as well. So she would like you to sort of deliberate on it further.
That is, of course, an excellent question, because I'm always asked this question. So in general, ligands do increase stability. However, there are a few cases where ligands actually destabilize and lead to an unfolding. In fact, you might have seen that one of the compounds actually had a destabilizing effect. And interestingly, we actually were able to solve the structure of that ligand with the destabilizing effect. And it turns out that this is a somewhat different binding pose. And in fact, this is a ligand that doesn't go right into the pocket, but a ligand that sits sort of at the end of the pocket. So it's almost solvent exposed. So in our case, we could actually show that the ligand that causes the decrease is a different binding pose or binding position. And in fact, it's a binding position that you wouldn't pursue because it's sort of on the edge of the binding pocket. It's still the inactive conformation of the protein, so it still has sort of the right effect, but it's really not what we would think of in terms of chemistry. So we did not pursue this. In other cases, I would say in at least 9 out of 10 cases, for other proteins, we see clearly an increase for ligands that we then further test. And in this case, we don't test activity, we just put them into crystallization right away. And but we have another case where we have an enzymatic assay, and we clearly see that the ligands that also work as an inhibitor for the enzymatic assay are usually the ones that have positive, significant increases in thermal temperature. So yes, it can happen, but normally we would try to avoid those.
Thank you very much, Dr. Pohl. So we have another question from Dr. Hassan. He's asking, normally people ask that finding new hits, that is in drug therapy, through VS, followed up pipeline, we might take already MDR or XDR strains even more resistant. We may make the already MDR XRD strains more resistant. In that case, what could be the ultimate solution to counter these foes?
So in terms of resistance, again, I should really give Alain Boulard, who is the researcher the senior researcher in Lille, credit for this. He has been working on actually this and a few others, ETHR, for decades. So the idea about using booster drugs is that you're actually using a completely different target. So you're not going after the same target again, by having a completely different target, you decrease the chances of mutations. I'm a chemist by training, but I'm enough of a biologist that I know that in the end, there will be drug resistance mutations coming up as well. And in fact, ETHR has been identified, the underlying gene for ETHR has been identified in drug resistant strains as well. Not very often, there's a few cases, but there are some cases. most of the drug-resistant mutations or ETH actually sits in the ETHA gene or in the INHA gene. So in the targets of the drug and in the targets of the prodrugs. So people have looked, of course, at mutations and most of them are there. But then again, there is a few mutations in ETHR as well. So I would say we can't win this race. We can only try to be one step ahead.
Thank you, Dr. Pohl. So there are so many questions here and we also have time. So, so there is one question from Mr. Anil Yusuf. He's asking, Professor, was it a fragment-based drug design?
What is a fragment-based drug design? What's the question?
Yeah, was it, I think the drug design that you have shown in your presentation,
yeah,
he's asking if that was. No,
and the reason is, I mean, we deliberately didn't do fragment-based drug design because our competitors in Cambridge, Tom Blundell, was working on fragment-based drug design already. So there's a sort of a parallel approach where they used a drug design, a fragment-based approach. So we deliberately did not do that because we really wanted to do something different. And also, when we started, there were already a few, I mean, again, 11 ligands out. So I think what we really tried to do is finding new chemistry in the Zinc database. So in this case, we deliberately did not use fragment-based screening. I should say that we have a different project, and I might explain this in Karachi in a couple of weeks. We have a second project where we actually employing fragment-based screening, and yeah, I might show this in a couple of weeks in Karachi.
Thank you. Thank you, Dr. Pohl. We have some other questions here as well. Did the team evaluate the in vitro or in vivo safety profile of the compounds in the clinical trial?
Yes, we didn't, and I wish we did. I can tell you that we wrote an EU application with Alain to do exactly that, and we came, as usual, we came one below the cutoff, so we didn't get the funding for this from a big EU proposal a few years ago, which was, of course, a big disappointment. But you're absolutely right, this is the next thing that we should be doing. Of course, the problem is that those tests become A, much more expensive, and B, are basically not fundable on sort of the normal basic research routes. So the only way, and again, Alain has done this with this company, the only way forward is finding a small company that is involved. But yes, I wish we could have done that, yeah.
Thank you very much, Professor Pohl. So here is another question from Mr. Ashish Arora. He's saying, great presentation, Dr. Pohl. Is 10 nanosecond simulation of protein ligand sufficient to derive hydrogen bonds and electrostatic interaction? And great to see the DSF results.
Yeah, and the answer is yes. We obviously we played with that. And I'm coming back to this one ligand that sits sort of on the side of the binding pocket. So if you see the binding pocket like here, and there's this one ligand that's sort of here, and all the other ones sit up here. So what we actually did is we started that simulation with the binding pose where the ligand is here, which is not the correct structure. And in fact, within the 10 nanoseconds, the ligand actually moved out by several angstrom and found the position in the X-ray crystal structure. So we were I was actually quite surprised how good the MD simulation is. So the answer is yes, it was surprisingly good. I would also say it's a relatively, I would say almost an easy target for MD simulations because the protein itself is actually really doesn't move a lot. There's this one phenylalanine on this side of the pocket that I showed that sort of swings around, but the rest of the binding pocket is actually quite rigid. So yeah, I think we were lucky with the target in many respects for method development.
Thank you very much, Professor. So we have another question, in which Joseph is asking, how were the in vitro or in vivo pharmacokinetics of the compound in phase 1 clinical trials?
So what was the question again? How, I mean, we haven't done, unfortunately, we haven't done that yet. So as far as we can go is, so the compounds are being tested in well, sort of in vitro and vivo assay, where we have attenuated Mycobacterium tuberculosis strains. This has been done in Lille, And we basically have growth curves with a reporter system that can be, so it's a GFP-based reporter system where we can basically get growth curves very conveniently. The problem, of course, is tuberculosis grows very slowly. So even those experiments take several weeks with each compound, and they have to be done with different concentrations of the ETH, the drug, in parallel. So basically, you test on how much do these drugs boost the efficacy of ETH. Yeah. So and again, we haven't done any safety profile yet. I would love to do that.
Okay, Professor, here's another question. What is your opinion on the factors that reduce time for a drug approval from 10 years to 2 years, as seen in COVID-19 vaccines and pills now?
I mean, the cynical answer is the expected profits from the company. Companies can do this very quickly if the expected profits are huge. And unfortunately, the expected profits for tuberculosis are not huge. So I think it really, and we know what we need to do, and we know that these tests can actually be done relatively quickly. So I'm afraid this is something where basically only private-public partnership can succeed. And there are some wonderful examples in the NTT field where private-public partnerships work actually quite well. Paul Denny, who might be here, might be able to answer how quickly miltefosine was went through the safety protocol. I think this was also 4 years. Maybe Paul is listening and can comment on that. So it can actually be done relatively quickly.
Thank you very much, Dr. Paul. This is the last question. Please tell us about how you choose specific residue that flips in the initial process.
So there's one more question I will answer, and that's repurposing, because that's one of my favorite ones. As I come to repurposing in a moment. I really think that having, well, somebody, scientist, a PhD student with a few people sitting on the screen and looking at the structure from all angles and really sort of turning around and ideally looking at more than one structure, a couple of ligand structures, you really get a good sense on what are the crucial interactions. So something that was very clear is, I haven't elaborated on this, but in all of these, there was a crucial interaction to a certain asparagine in the center of 179. And actually, it's the one that's been turned around. So we really realized that this is a crucial interaction for all of the ligands. And in fact, having this hydrogen bond was something we put into the very early pre-screening protocol up there. But we also realized that some other residues, like this phenylalanine on the side 184, can actually be turned around. So from the crystal structures, we knew that there's a flexibility. And I think that's another important take-home message, is that one crystal structure gives you a beautiful picture and gives you a ribbon diagram and so on. But it's only multiple structures that give you the detailed information, for example, about the flexibility. Which residues can turn around, are likely to turn around, which residues never turn around. And that's something, again, you learn from analyzing multiple crystal structures. And when I say analyzing, looking at these crystal structures. And I mean, my colleagues know this. I do spend quite a lot of time just on my screen looking at crystal structures and turning them around and sort of making sense out of that. I would like to answer the question about repurposing, because there's been a huge discussion, particularly with the COVID-19, about repurposing drugs. And I can tell you that I've been asked to review some research grants for repurposing. I'm not a big fan of repurposing, and I think actually if you look into the detail of repurposed drugs, there's very few, if any, examples where repurposing without any hypothesis has worked. And in fact, I would claim that the few cases where repurposing sort of worked were actually based on clear scientific understanding. And simply throwing drugs onto a target or onto a disease without any hypothesis is actually not going to work. So I would say repurposing, yes, if there is a clear scientific understanding and a hypothesis that can be tested. I'm probably a bit more on the skeptical side with that.
Thank you very much, Professor Pohl. And again, cannot thank you enough for your time and for such a wonderful presentation. You can see so many comments regarding your presentation as well. And we hope to see you in Pakistan someday as well, inshallah, in Islamabad particularly.
I know. Thank you so much. That was really good, and I really appreciate the comments. I mean, you guys, you're too kind.
Thank you. And for all our participants, this lecture will also be available on YouTube. So you can go on our YouTube channel and you can go through this lecture once again if you would like to. And again, thank you very much to all of you for attending and having a very interactive session and commenting on this, for being very generous with your feedback. And thank you very much, and hope to see you again, inshallah, and bye-bye.